Before we start, I'd like to thank Horiba Scientific for supporting this session. And without further ado, I will just pass it on to Ochiya Sensei to introduce Dr. Koji Ueda. Please go ahead, Ochiya Sensei.
Okay, thank you very much, Carol Nelson. And then I'm very happy to introduce our speaker, Dr. Koji Ueda, project leader of Cancer Prevention Medicine Center, Japanese Foundation for Cancer Research, Tokyo, Japan. And if I briefly introduce his biography, in 2008, he got a PhD at the University of Tokyo. And then he got a position at the RIKEN, which is a very famous Japanese Institute of Physical and Chemical Research. Then he stayed for 6 years. And then he moved to the current position, Japanese Foundation for Cancer Research. And his background is, proteome analysis. And of course, he's very familiar with genome analysis, of course, and mixed research, I guess. And then he belonged to the different societies and different groups, and he working on the editor board membership and also associate editors of some very famous international journals. And he's a counselor of Japanese Cancer Association, and also he is a member of the ICEP, of course, and also he's a board member of our Japanese Society for Extracellular Vesicles. And his many awards in 2018, he got an award for the Young Investigator of the Japanese Cancer Associations. And today, of course, he's one of the, leadership on the tissue-derived EVs analysis. And currently, he's most focusing on the biomarker analysis based on the EV proteins, and of course, many other aspects. But we're very much happy to know his current progress on these areas. Okay, Koji, are you ready?
Okay, so actually, I'm very happy to have this great opportunity to have my presentation about the UV biomarkers for cancer in this session. So this is the brief introduction of our institute, Japanese Foundation for Cancer Research. Actually, this institute have the research center and also the cancer-specific hospital. And actually, the number of the population-based cancer registry is #1 in Japan among all the cancer-specific hospital in Japan. So now we have actually many resources of the biological specimens from the hospital, and we have also the very good research institute next to the hospital. Then recently, you may remember, actually our institute was really surrounded by the Olympic game centers and parks. This is our institute in Ariake, the Bay Area of the Tokyo. And around this area, we have many Tokyo Olympic Games, including the tennis and urban sports, and many water sports, small sports. And just in front of my institute, the Tokyo big site was used for the worldwide press center for the Olympic game. We hope you enjoyed the Tokyo 2020 Olympic and Paralympic Games. Please remember also our institute. Then in my group, now we are working on the development of new technology for the cancer detection or cancer problem diagnosis based on the proteome analysis technologies, especially using the mass spectrometries. Then today, of course, we would like to talk about our researches for the exosomal biomarkers. Then, as all of you know, the exosomes are basically secreted from all of the human body cells and can be detectable from blood or CSF or maybe urine. And the function is also very well known for cancer cell-derived exosomes, including the promotion of the angiogenesis or the immunoediting. From the function of the DNA, RNA, proteins, and metabolites encapsulated into the exosomes derived from cancer cell. And especially for the diagnosis, now we are developing many different technologies for the exosomal diagnosis for the liquid biopsy of the cancer. Now, for the cancer liquid biopsy diagnosis, we have mainly 3 ways, the CTCs and also the ctDNA, and one of them should be the exosome. Then, the ctDNA diagnosis is already used in clinical situations, including the foundation 1 liquid biopsy in clinic. But now for the exosomal diagnosis, many companies are planning to develop their own technologies, own products for the diagnosis for cancer and many other diseases. For example, the exosome RNA, urinary exosomal RNA diagnosis kit for the detection of the prostate cancer from 2 companies. And for example, some of them are using the new technologies. For example, the aptamer specifically bind to the cancer-derived exosomes, and in this case, they use the new exosome isolation tool, hem purifier. And actually, these are assigned as the FDA breakthrough devices. So like these ones, the exosome-based diagnosis are already in the production by the, several companies worldwide. Then in our case, we started to establish our own strategy to isolation of the exosomes from serum or plasma samples at first stage. Because now we have many options for the purification and the isolation of exosomes from biological samples, typically using the virtual centrifugation and many other commercial kits. But especially for our case, I mean the case of biomarker discovery using around 100 of samples, it's quite difficult to secure the reproducibility, and we need much time to perform the preparation of samples before mass spectrometry analysis. And actually, the purity of the exosomes is not sufficient for the mass spec, at least for the proteomics analysis. So we aim to develop our own technology to acquire high purity exosome from the serum plasma samples. And this is the first one, the MSIA initiative by collaboration with Sam Fisher Company. We, in this case, use the monolithic silica filter at the tip, at the edge of the tips. And so we bound the anti-CD9 antibody to capture the exosomes at the edge of the tips. And using this chip, we can operate with the robot, and we can isolate the exosome samples for 96 samples simultaneously in only 30 minutes. And this product was actually finally commercialized from the Cosmo Bio company as the Exotrap spincoram. So we use this type of spincoram and easily isolate, immunoprecipitate the CD9 positive exosomes from any type of liquid samples using simple spin. The next one is EB2 coram. This is based on the size exclusion chromatography and also the weak hydrophobic interaction. But the preparation is quite easy without any specific reagents and without any specific machines. With only simple gravity flow, we can obtain very high purity exosomes using this coram. For example, as shown in this graph, we can correct the high purity exosomes in this fraction, independent from the serum abundant proteins. So this coram was also commercialized by Geoscience International company. And now probably this is available worldwide. And the third one is was developed by the Professor Hanayama in Kanazawa University in Japan. And this kit is also useful for the high purity exosome isolation. This is based on the phosphatidylserine serine specific capture by the magnetic beads. So we can operate the magnetic beads robot or operation robot, and we can isolate the very high purity exosome also. Then anyway, for example of the exosome proteomics analysis using the EB2 coram. In this case, we purified exosome from 58 serum samples, basically, so including the healthy donors and the gastric cancer patients. And from this proteome analysis, interestingly, we identified the pathogenic protein CAG-A. This is the pathogenic protein from Helicobacter pylori. So we identified this protein from the gastric cancer patient serum exosomes. So now we think this is one of the causes of the autoimmune diseases and also the other extra gastric diseases caused by the infection of the Helicobacter pylori. So, and also for the basic insight from this analysis, we observed many interesting relationships between the exosome marker proteins. For example, the tetraspanin 9 molecules and the CD9 molecules show a very high correlation score for the expression level. But for example, the CD81 showed had quite low correlation with the other tetraspanin molecules on these 60 samples. So this is also important knowledge when, for example, when we use these molecules as the standard or the normalization standard in the any experiments, we should select the appropriate Z+ binding molecule for the normalization, I guess. Then actually in human body, so far 33 Z+ binding genes are known. So now in usual proteomic study for the exosome, my laboratory is verifying the exosome purification score. So this is the calculation method for our original exosome enrichment score to secure to know the exosome purification efficiency for all the samples. And the concept is very simple. We can calculate the score by the abundances. That means the total expression level of the Z+ binding molecules. In this data set, we detected 13 Z+ binding molecules. Then we calculate the total number of the Z+ binding molecules, and finally calculate the ratio of this one with the sum of all detected proteins from the exosome samples. In this case, the score was 0.15%. This is very simple but useful for the verification of the purification efficiency of the exosomes. This is example of the validation of the exosome enrichment score from different exosome isolation technologies. The ultrafiltration centrifugation and X-quick score, the score was 0 actually, and the CD9 antibody affinity and EV seconds size exclusion chromatography and the mag capture, phosphatidylserine based magnetic bead capture technology. The red colored numbers are the exosome enrichment score. You can see there are these scores from the size exclusion column and the magnetic beads isolation were very high compared to the orbital centrifugation and other technologies. Like that, for example, when we performed biomarker, biomarker screening, biomarker discovery experiment using around 100 or more than 100 samples, we calculate this score for each sample and validate the exosome purity efficiency for all samples and select the appropriate sample for the next statistical analysis. But today I'd like to show you the new strategy now we are using for the biomarker study for cancer. The cancer-derived exosomes isolated from the viable tissue samples. We can do that because we can tightly correlate with the hospital. Actually, the 20 operation rooms are only 50 meters distance from my laboratory on the same floor. So the first example is the biomarker discovery using that technology for kidney cancer. In Japan, the incidence and the number of incidence and the mortality of the renal cell carcinoma 2 times or 2.5 times higher than the 2000. Very pleasing, but now we have no specific biomarkers for kidney cancer. So we aim to identify the exosomal biomarker for kidney cancer using this technology, TEEP. In this case, we collected the tumor region and also the control region, the normal region, from the same the surgically resected tissue in the operation room into the serum-free medium. And after collecting this viable tissue, we go back to my laboratory and start incubation for several hours. Then the secreted exosomes were isolated, enriched, and performed proteomic analysis, and maybe in some cases other type of omics studies. We may name this one tissue-derived extracellular vesicles TIV. So this is very useful resource for the biomarker. The TIV do not include the serum abundant proteins because we use the serum-free medium, and we can directly collect the viable tumor tissue-derived exosomes only. That means without whole body derived exosomes. And the next one is also important for the comparative study. We can collect the normal and tissue paired samples from the genetically identical samples. And the next one is also interesting feature of this technology. We can amplify the biomarker results by the elongated incubation time. So from only very small amount of samples, like biopsy samples, we can collect the enough sample amount, enough exosomes by the prolonged incubation. So anyway, using these are actual pictures of the incubation of the viable tissues after several hours. Then using these T samples from 20 patients, finally we identified in this case around 4,000 proteins, and fortunately identified the adenosine as one protein specifically expressed on the tumor lesion-derived exosomes. The fold change was actually more than 30. Then in this analysis, we observed more than 7 orders of very wide range of 4,000 proteins in exosomes from the viable tissue. And actually, the astrocytin is known as a membrane-bound protease protein, around 20 kilodalton. Then this is a picture of the electron microscopy of the exosomes derived from the viable tissue. And you can see the expression of the astrocytin on the surface of the exosomes like this. Then importantly, we can detect the exosomal astrocytin also from the serum samples collected from the kidney cancer patients. And importantly, when we construct the exosome sandwich ELISA analysis using the the anti-CD63 antibody and the astrocytin antibody sandwich system, we observed a clearly kidney cancer stage-dependent expression profile on the exosome only from the 10 microliter of the serum. So probably we believe this protein is quite useful for the detection of the kidney cancer using small volume of the serum samples. And in this analysis, in this study, we also analyzed several molecular function of the exosomal astrocytin protein. When we incorporate the labeled exosomes into the HUBEC cells, we observed the incorporated exosomes like this. And interestingly, when we prepared the eliciting overexpressing exosomes and applied them, route them to the Hubeck seat and perform the transwell cell permeability assay. We observed significantly destroyed collapsed structure of the Hubeck cell sheet after the incorporation of the eliciting positive EVs like this after 12 or 24 hours. And finally, we observed significantly increased invasion of the kidney cancer cells through the Hubeck cell sheet after the preparation of the eliciting strongly positive EVs treatment. So we hypothesized that eliciting strongly positive EVs from the kidney cancer cells may affect for the hematogenous metastasis of the kidney cancer cells in human body. Now we are validating this result using in vivo experiment. And for biomarker application, we are now collaborating with Japanese diagnostic company, Toso Corporation, and using their high-speed ELISA system, we are constructing the kit to detect the exosomal isoleucine from the serum samples. Of course, this type of preparation of the TIV samples is applicable for any type of solid tumors. So we recently applied this strategy also for the colorectal cancer biomarker analysis. So in this case, in addition to the analysis of the exosome samples, we also analyzed the tissue proteomes and compared the expression profiles. Similar with the kidney cancer case, we collect the tumor region and the normal region of the colon tissues into the serum-free medium from 17 patients. We collect the TIV samples and also the tissue samples, original tissue samples, and performed proteome analysis. In this case, in total, 10,000 proteins were identified, and more than 6,000 exosomal proteins were identified and quantified. This number was actually very huge. And when we compared this number with the XCART database, indeed, more than 2,600 proteins were newly identified in this study, not shown in XCART database. So, and also interestingly, we compared the expression profile between the exosome proteins and the original tissue proteins. We found very low correlation between these two proteome profile. That means probably there are very specific mechanism of the protein incorporation into the RODS mechanism into the exosomes from the original cell. Then from this big data set, interestingly, we identified new UV biomarkers, not the membrane protein, UV luminal protein biomarker, VPS family. We identified actually 12 VPS family proteins, and as shown in this box plot, all of these proteins were highly regulated and enriched in the exosome samples compared to the original tissue samples. This is very similar with the tetraspiny molecule profiles. So these proteins should be useful for the marker protein, EV marker protein for the luminal ones. Then for biomarker discovery analysis, after the several statistical analysis, we fortunately identified this protein, the CAT1, cationic amino acid transporter 1, a much past membrane protein. This is actually the transporter of the amino acid, especially for the cationic amino acid like arginine. This protein was found to be express very specifically on the colorectal cancer tissue-derived exosomes, and very low level in the normal colon tissue-derived exosomes like this. And then when we construct again the exosome sandwich ELISA system, in this case we use the sandwich system with CD81 antibody and the CAT1 antibody. Then measured the exosomal CAT1 level from the 10 microliter of plasma sample, more than 100 of the samples from stage 1 to stage 4 colorectal cancer patients and the normal healthy donors. And importantly, we detected significantly higher level of the exosomal CAT1 level even in the very early stage of the colorectal cancer. The AUC was 0.78. Sorry, this one 0.82. And when we combine with the CEA, the traditional colorectal cancer biomarker, the sensitivity and the specificity was slightly improved, and the AUC was 0.87. But anyway, among many candidate proteins, now we believe this protein is very useful for the early detection of the colorectal cancer from the plasma sample. And again, for the functional analysis of this protein, we found the relationship between the angiogenesis by this protein activity. So we constructed the CAT1 positive AVs, and again, we used the Hubeck cell system. And in this case, after the incorporation of the CAT1 positive exosomes into the Hubeck cell sheet, we observed significantly upregulated cell growth of the Hubeck cells, and also the upregulated migration speed of the Hubeck tubes and to the 3D culture system. This result means that the upregulation of the Hubeck derived the tube formation promotion. So this is a very important knowledge for the relationship between the angiogenesis. And we also performed the metabolomic analysis after the incorporation of the CAT1 positive exosomes into the Hubeck cells. And when we performed the metabolomic analysis around the pathway of the NO, the nitric oxide metabolic pathway, we found that finally the arginine from the arginine transportation significantly upregulation of the cyclic GMP was observed after the upregulation of the CAT1 protein on the Hubeck cells. So this is very well known phenomenon in the angiogenesis using as shown in this picture. After the upregulation of the NO cycle and the upregulation of the cyclic GMP, this finally introduced the upregulation of the MAP kinase pathways, especially for the ARC, and also the FAK to promote the proliferation and the micrylation of the vascular endothelial cells, and finally induce the angiogenesis in the microenvironment of the tumors. So like this, from the viable tissue-derived exosomes, we identified many biomarker candidates, and now we are validating these results using the more number of human samples with the company-based diagnostic tools like this picture. But anyway, so our strategy, I mean, the viable tissue-derived exosomes should be very useful resource of the biomarker discovery for any other type of tumor samples. Then we are actually very happy to collaborate with some of you using our hospitals, this very precious kind of samples for the biomarker discovery and validation. Thank you very much. Thank you very much for attention.
Okay, thank you very much, Koji. It's a very nice talk, as always. And maybe you have accepted a question from the audience. Okay, oh, Hector, how are you? Hello. And okay, thanks, Koji. A really nice approach. Have you tested whether AZU cat segregated exclusively by tumor cells or by any other cell types in the tumor mass? Koji, would you respond to Hector's question?
Sorry, it's difficult to hear. Could you repeat the question?
All right. The Hector asking whether you tested AZU cat secreted exclusively by tumor cells or any other type of cells in the tumor.
Okay. In our case, we actually collected the tumor. Sorry.
Yeah, it's okay. Go ahead. Go ahead.
All right.
Hector is appeared.
So we corrected the tumor lesion and then also for the control lesion. And so after collecting the tumor samples, we validated the tumor contents of our samples with a pathologist. And basically, we believe so these exosomes are derived from tumor cells mainly, but of course, the tumor cell, the tissue samples may include many other, for example, the stromal cells and the vascular endothelial cells. So it's quite difficult to separate the exosomes among these cells.
Hector,
yeah, Koji, yes, definitely. Yeah, actually, this is a trouble we have ourselves in our approaches, because one of the limitations of our approaches is that since the tumor mass is composed by probably 50% of stromal cells, it could be a limitation of anyone, yes, including myself. But of course, my question is, if you tested, for example, cell lines, tumor cell lines, or endothelial cells, if they express your protein, and if this could be, of course, in vitro, can you achieve or envision if this is just tumor, or it could be both, it could be tumor and endothelial cells. I don't know if you play a little bit with the system in order to see if this is only tumor or it could be also endothelial cells, for example.
Yeah, that's very nice idea to confirm our results. So, but unfortunately, we did not yet. We did such kind of we did not perform such kind of experiment to compare the in vitro the cell line derived exosomes and the tumor or tissue derived exosomes. But yeah, as you said, when we perform the quantitative comparison between the many other so 100% of tumor cell samples with the cancer tissue samples, we can obtain many important information from the tissue samples. And yeah, it's important knowledge base, I believe.
Yeah, it's easy, you know, because we have faced several troubles with, of course, endothelial cells in a tumor mass are different than in the normal tissue. So I don't know, if you take, it's challenging to isolate endothelial cells from tumors because, you know, putting them in plastic could achieve that. You know, maybe these markers that you have are also somehow by tumor endothelial surrounding cells. So this could be a transformation marker for the endothelial. So in other words, in your HUVECs are recepting these proteins, maybe it's because they are affected by exogenous material and could be transformed by the tumor. So I see an angle, you know, there is an angle maybe if you can block this in endothelial cells and tumor cells, it would somehow, you know, prevent tumorigenesis or at least control a little bit. But you know, thank you so much for your talk. I really enjoy it. Of course, the technology is really good. You know, I would love to have your connection with the clinics because you have this ELISA assays that is right into the clinic. So it's really awesome what you guys are doing. So congratulations for your work.
Yeah, thank you very much. Actually, now we are analyzing the pancreatic cancer samples also using this technology. But as you know, the pancreatic cancer tissue includes many, so much volume of the stromal cells. So probably in that case, that type of cancer samples, it's difficult. It's important to so obtain the cancer cell line information also. So thank you very much.
Okay, thank you.
Okay, thank you, Hector. Okay, next question is from Cecilia. Cecilia, hello, are you there? If you're there, so please.
Yes, I'm here.
Yeah, say please make a question for Koji. It's
thank you for a very nice presentation. As Hector highlighted, it's a very cool technique. I was wondering, did you find ASU1 in colorectal tumor EVs or CAT1 in the kidney tumor EVs? Did you see like a good specificity for these markers, and have you tested them in other types of cancer, maybe in this pancreatic cancer that you mentioned now?
Yeah, actually, we identified the AS1 from the kidney cancer and the CAT1 from the colorectal cancer. And so far we are collecting the EV sample, the T samples from many other types of cancer, solid tumors in our hospital. But so far now we have no data set by comparing that biomarkers from the different type of tumor samples so far.
Yeah, because I guess when you have this nice ELISA system and you use just a very little of the serum samples, you could cross-test from the different patient groups to see the specificity.
Possible. Yes, we will do that.
Great, thank you.
Okay, thank you, Cecilia. Okay, next question is from Clara, and she's asking about the, you know, there's some technical aspect for the protein purification EVs. Clara, hello, are you there? You. Would you please say directly to Koji? Great question, thank you.
Thank you. It was just a technical question. Considering the low yield of the recovery of extracellular vesicles protein, if you suggest to perform, for example, some fractionation after the trypsin digestion, or for example, a double digestion with trypsin and another enzyme, if you suggest this, or you, in your experience, is better not to manipulate more the sample. Thank you.
In our case, we are using the proteomic analysis. So it's important to acquire very high purity exosome. And for all the purification strategies from the serum, plasma, urine samples, Finally, so far we use the fractionation, size exclusion chromatography fractionation, and also the phosphatidylserine specific magnetic beads capture protocol to obtain the very high purity exosomes from these samples. And so as you know, so the purity of these exosomes are quite important for the final result of the proteome analysis and many other type of omics analysis. So when we use the serum plasma samples, now we are using the two type of purification technologies.
Okay, thank you.
All right, thank you, Clara. Okay, and please write down, or maybe you open up your windows and talk to directory. Oh, another question. Okay, Carolina-san. All right, please.
Yes, I'll take this opportunity to ask question. Thank you so much, Koji. It's very interesting and impressive data that you presented today. I'm really interested with the finding that you shown that you found a lot of VPS protein, which is partly they are at least some of them are shown to be involved in the exosomes formation. So I was just wondering whether this is sort of, found in different cancer data sets as well, in other cancers, and whether they are potentially useful to be used as the proteins to measure exosome purification, like the one that you shown with the tetraspanin for the exosomes enrichment score.
Yeah, thank you so much for the important question. For VPS family, so actually from our many mass spectrometric analysis data set from the different type of cancer samples, we also detected very specific enrichment of the expression of the VPS family in the exosomes compared to the total cell lysate analysis. So we believe these proteins can be used for the universal marker for the luminal exosome. Yeah.
Yeah. Have you looked into a different cancer? Are they also found in abundance in different cancers?
Yeah. At least we have the data set for the kidney cancer, colorectal cancer, prostate cancer, and pancreatic cancer. From all of the data sets, we detected very high level of the VPS families, like lab family proteins in exosome.
That's pretty cool. Also, it's quite interesting the data that you shown about the using the exosome enrichment score, that it looks like the magnetic capture is actually provide the best purification.
Yes.
Am I getting it right? So you sort of like using phosphatidylserine specificity to isolate them, and that sort of performs better than CD9 or tetraspanin capture?
Yeah, in our result, that's true.
Oh, that's cool.
Yeah, but of course, this type of experiment, the exosome enrichment score is strongly dependent on the strategy, the mechanism of the purification, and now we have many different options for isolation of the exosomes, and it's important to important to calculate this score by yourself in each laboratory. Probably this score, the number of this score should be quite different among the laboratory, I guess. So, but in our situation, using the magnetic beads and also the size exclusion chromatography were the best score.
Okay, thank you. Okay, so okay, Koji's relation to your very fantastic EV second technology. So have you ever checked, because I said mentioned the categories of EVs as a small EV or middle EV, large EVs and something, right? So is it possible to separate all those categorized EVs by your EV second?
Yeah, I'm not sure the definition of the small and large EVs, but we did not perform such kind of experiment. But basically, in the principle, we can fractionate the extracellular vesicles into different sizes, into the different fractions from the size exclusion chromatography. So if we perform the more detailed fractionation and perform the proteomics and other particle nano-tracking analysis, maybe we can observe the difference of the more molecular features in the different fractions, and also the shape of the EVs from the size exclusion chromatography of our EV second. But we did not perform such kind of
that's good. Yeah, it's fine. Yet. Yeah, fine. Yes. My another question is, of course, you know, we are the cancer researchers, so the priority is cancer. But of course, as many people working on the tissue architecture organoids. So the organoids is maybe they're producing extracellular vesicles from different type of tissues. So is it possible to apply your idea or lab tissue EB analysis to those organoid culture?
Basically, that's possible. And for example, we, in addition to the cancer samples, now we are collaborating with the people studying about the neurodegenerative diseases, Alzheimer's disease and Parkinson's diseases. So they have very nice mice model for that type of diseases. And so we are now trying to isolate the brain sample from the mice sample and the spinal cord in some cases. And from that viable, the fresh tissue samples of the mice, we succeeded to isolate the APs. And now we are analyzing such kind of samples also from the disease mouse and the normal mice. Okay. So basically, any type of diseases, we can use this type of experiments if the tissue samples are available.
All right. How about the tissue slice? Okay, sometimes people working on the brain tissue slice and they put on the culture and then analyze for them. Is it possible to do that?
If
tissue slice.
If the cells are viable,
of course, that's the point.
Yeah.
Okay. Yeah.
We can isolate.
Yeah. If the tissue slice is viable, and then if you trace the EVs, and you can mapping on the different EV characterization, and so maybe it's quite interesting. And that's simple.
Yes, that sounds good.
All right. Yeah. Thank you. So how about the audience? Another question for Koji? Is that all? Okay, so now seems to finish up. So I hand my hand to Caroline-san.
Yeah, thank you so much, Ochiya Sensei, for leading the QA. It's a wonderful session. And also Koji, again, wonderful presentation today.